Myself, Dipali Shelke, welcome you all to this presentation on classification of crude drugs. After completion of this topic, students will be able to define crude drugs and classify it, identify the different types of crude drugs, and describe the points used to study the details of medicinal plants. Also categorize the term related to the biological and geographical origin of crude drugs, and apply theoretical knowledge to practical. Crude drugs. Crude drugs are nothing but the products of plants or animal origin found in the raw form, or the pharmaceutical products from the natural origin, and the substances obtained from the mineral origin like kaolin and bentonite. Crude drugs are further classified as organized and unorganized form. Organized means having the definite shape and size, and unorganized means they do not having any shape and size. Now it's time to think and reflect. Pause the video and record your answer in notebook on a question: what is the need of crude drugs classification? If you have finished writing, play the video and see the answer. Let me answer you that why there is need of classification, because to identify them into different groups according to their botanical names, also to manage the data, also to remember each plant species according to their names, and to avoid the confusion. Hence, there is a need of crude drug classification. These are the different types of methods of classification of crude drugs. First one is the alphabetical, second one is taxonomical, third one is the morphological, fourth one is the chemical, fifth one is the pharmacological, sixth one is the chemotaxonomical classification. We will see each one by one. First one is the alphabetical classification. In this classification, crude drugs are basically classified according to their Latin or English name and arrange them according to their alphabets. For example, they are arranged in the form of alphabets: Indian Pharmacopoeia, British Pharmacopoeia, United States Pharmacopoeia, as well as National Formulary. In British Herbal Pharmacopoeia, in British Pharmaceutical Codex, for example, such as acacia, benzoin, cinchona, deal, ergot, fennel. Next method is the taxonomical classification, in which the drugs classify according to the plants and animals from which they are obtained, such as phyla, orders, families, genera, species, subspecies. This system is first to recognize the drug, like organized or unorganized nature of the crude drug, as well as their chemical nature, as well as the therapeutic action of the crude drugs. For example, the glycyrrhiza drug. It included in the phylum Spermatophyta, division Angiospermae, class Dicotyledon, order Rosales, family Leguminosae, subfamily Papilionaceae, genus Glycyrrhiza, and species Glycyrrhiza glabra. Next method is the morphological classification. Drugs classified according to their part of plant, as well as describe them into organized and unorganized drugs. For example, such as the part of plant is the seed. Drugs included the Nux vomica and Strophanthus. For leaves, Senna and digitalis. For barks, Cinchona and Curci. For woods, Quassia, and roots, Rauwolfia, and so on. Next method is the chemical classification. Crude drugs classify according to the chemical nature of active constituent of the plant species. First category is the glycoside. Included the drugs digitalis, senna, cascara, and licorice. Alkaloids included the Nux vomica, ergot, cinchona, and datura, and so on. And it is the most preferred method of classification, since their therapeutic as well as the significance is based upon the chemical classification of the crude drugs. Next method is the pharmacological classification. In this, the drugs classify according to the pharmacological action of active constituent of the crude drugs and their uses. Drugs having the different mode of action but having the same pharmacological action are grouped in one group. For example, purgative. Purgatives are again classified into three types: bulk purgative, irritant purgative, and emulient purgative. For example, drugs acting on GI system. First category is the beta, second carminative, third emetics, and fourth one is the anti-amoebiasis. In beta category, it included the drug gentian, quassia, cinchona, and in carminative category, it included the drug dill, mentha, and cardamom. Likewise, in emetics, ipecacuanha, and in amoebiasis, curcumin and ipecacuanha, and so on. Chemotaxonomic classification method included the combination of both chemistry as well as taxonomy. There are 20 metabolites having the pharmaceutical significance, such as alkaloid, glycosides, and flavonoids. It is hard, such as DNA having the significance in hybridization, amino acids sequencing in a protein. Likewise, serotaxonomy also gaining the significance in this method of classification. For example, presence of berberine alkaloid in the berberries, presence of rutin flavonoid in the flavonoids containing in the higher plants. These are the different methods of classification of fruit drugs. After studying this, students will be able to define all the methods of classification and able to understand the classification of fruit drugs. Thank you.